Hi friends, welcome to our channel Knowledge Rain. Today we are going to discuss a new set of question paper and this is MCQ part 6. Let's see. First question. Identify the sugar with free aldehyde or ketone group. Identify the sugar with free aldehyde or ketone group. The options are option A glucose, option B sucrose, option C pectin and option D cellulose. Identify the sugar with free aldehyde or ketone group. A sugar with a free aldehyde or ketone group is usually known as reducing sugar. Reducing sugar is any sugar that is capable of acting as a reducing agent because it has a free aldehyde or a free ketone group. So here the answer is glucose. Glucose is a sugar with free aldehyde group and the sugar with free ketone group is fructose. Almost all the monosaccharides act as a reducing agent or a reducing sugar. Next question. The brown color of bread crust is due to. The brown color of bread crust is due to. Options are caramelization, Maillard's browning, enzymatic browning and Strecker reaction. The brown color of bread crust is due to Maillard's browning. What is this Maillard's reaction? It is a chemical reaction between the amino acids and reducing sugars. It is a chemical reaction between the amino acid and reducing sugars which will give brown food its distinct flavor and color. It will induce a brown color. It is due to the reaction of amino acid with the reducing sugar. It gives a brown color and a distinct flavor for the food. So the brown color of bread crust is due to Maillard's browning. And let's see what is caramelization. Caramelization and Maillard's browning comes under non-enzymatic browning. Here the caramelization is nothing but the browning of sugar. And this results, the caramelization results a brown color and a sweet nutty flavor. And next one is the enzymatic browning. And this occurs in fruits and vegetables, usually resulting in negative effects on color, taste and flavor. And this reaction is a consequence of the phenolic compounds and oxidation of that phenolic compounds by polyphenol oxidase. And this triggers the generation of dark pigments, that is enzymatic browning. Next one is Strecker reaction. Strecker reaction is nothing but a method of synthesis of amino acid. It is nothing but a method of synthesis of amino acid. Next question. Deposition of fat within lean muscle of meat is known as. Deposition of fat within lean muscle of meat is known as. The options are stunning, hoisting, tenderizing and marbling. Deposition of fat within lean muscle of meat is known as marbling. What is marbling? That is a high quality steak or a piece of meat will have a lot of marbling. That is while a lean cut will have very little or no invisible marbling. That is, if we cut a steak or if we took a steak of meat, we can see some patches of white patches here and there. That patching is known as marbling. It is due to the presence of fat. That is, the deposition of fat within a lean muscle of meat is known as marbling. And what is this stunning? A stunning is we already discussed the term stunning in last video regarding adult cattle slaughtering. The best stunning method suitable for adult cattle slaughter is captive pistol blot. Here the stunning is nothing, the process of rendering animals immobile or unconscious with or without killing the animal. and when or immediately prior to slaughtering them for food. That is, immediately before slaughtering, rendering the animal immobile or unconscious is known as stunning. And tenderization. Tenderization is the technique for breaking down collagens in meat to make it more palatable and tender or soft are referred to as tenderization. That is the technique of making or breaking down the collagens in meat to make it more palatable and soft is known as tenderization. In this slide, the answers are marked. And coming to next question, which of the following helps in transport of lipids in the body? Which of the following helps in transport of lipids in the body? Options are myoglobin, keratin, albumin and lipoprotein. The answer is lipoprotein. Lipoprotein helps in transport of lipids in the body. Why we need this lipoprotein? Cholesterols or the lipids are minimally soluble in water. They are non-polar, so they are insolvent in water or minimal solubility in water. So it cannot dissolve and travel in the water-based bloodstream. Instead, it is transported in the bloodstream by these lipoproteins, which are water soluble and they can carry the cholesterol and triglycerides internally. And they carry the fat mostly as triglycerides. And this is the action of lipoproteins or the role of lipoproteins. Lipoproteins helps in the transport of lipids in the body. As the lipids are insoluble in the water-based bloodstream, lipoproteins help the lipids to transport in the body. Next question. Function of vitamin B complex is as the function of vitamin B complex is as enzyme, antioxidant, coagulant or coenzyme. The answer is coenzyme. The function of vitamin B complex is as coenzymes. What is difference between a cofactor and a coenzyme? That is, the non-protein component of certain enzymes are called as cofactors. But if the cofactor is organic, then it is called coenzymes. The coenzymes are relatively small molecules compared to the protein part of that enzyme, and of course many of the coenzymes are derived from vitamins. The coenzymes make up a part of the active site, since without the coenzyme the enzyme will not function. So the function of vitamin B complex is mainly as a coenzyme. Next question. Saponification value of a fat allow us to compare. Saponification value of a fat allow us to compare. The options are degree of unsaturation of fatty acids, number of phosphate group in a molecule, average chain length of fatty acids, and number of hydroxyl groups present. The answer is average chain length of fatty acids. Saponification value of a fat allow us to compare average chain length of fatty acids. What is the saponification value? Saponification value represents the number of milligrams of potassium hydroxide required to saponify 1 gram of fat under the condition specified. Under a specific condition, the amount of milligram of or the number of milligram of potassium hydroxide required to saponify 1 gram of fat is known as saponification value. And it is a measure of, that is, the saponification value is a measure of average molecular weight of fatty acids or the chain length of fatty acids. So saponification value is noted to get the average chain length or molecular weight of the fatty acids. And degree of unsaturation of fatty acid is measured by degree of unsaturation of fatty acid is measured by knowing the iodine value. Iodine value will give the total unsaturation of oils or fats present, or the degree of unsaturation we will get through iodine value. Next question. A person has normal vision in bright conditions but cannot see clearly in dim light. Which of the following vegetables is most effective in helping him to overcome the problem? The options are broad bean, potato, spinach, and green pepper. Broad bean, potato, spinach, and green pepper. Of course, the answer will be spinach, as spinach is a rich source of vitamin A. We already know that vitamin A is very much needed for a vision. A good vision, vitamin A is needed. So here the answer is spinach. Next question. The bright cherry red color of cut meat is due to. The bright cherry red color of cut meat is due to. Options are oxymyoglobin, metmyoglobin, myoglobin, and myoglobulin. The bright cherry red color of a cut meat is due to oxymyoglobin. That is, this color occurs several minutes after cutting and after exposure to oxygen. After exposure to oxygen, the meat will have a bright cherry red color due to the formation of oxymyoglobin. If that meat has a purplish red color, it is due to myoglobin. And if it is brown color, it is due to metmyoglobin. And these are the different colors of a cut meat. If it have a bright cherry red color, it is due to oxymyoglobin. If it is having a brown color, it is due to metmyoglobin. And if it is a purplish red, it is due to the presence of myoglobin. Next question. Richest source of iron is papaya, mango, pomegranate, and dry carota. Richest source of iron from this group is dry carota. Dry carota is having a 39.1% of iron in it. The richest source of iron is dry carota. Next question. Hot break system is followed in tomato processing to deactivate. Hot break system is followed in tomato processing to deactivate pectinase, pectic methyl esterase, protopectinase, and polygalactourinase. Hot break system is followed in tomato processing to deactivate pectic methyl esterase. Pectic methyl esterase. Hot break method, which involves processing temperature of 90 degrees Celsius or higher. There is another method followed in tomato processing, which is cold break method, and it has a processing temperature of 77 degrees Celsius or lower than that. And the hot break system, which will help us to get a more viscous product, and this system is followed for the preparation of ketchup, sauces, and pastes. And while in cold break, the product will have a better flavor and color retention, and it is used for less viscous products such as tomato juice. If it is for juice purpose, we will use cold break system, and if it is for a sauce, ketchup, and paste, we will use hot break system. That is for high viscous products, hot break system, and for low viscous product, cold break system. Here the question is, hot break system deactivate which enzyme? The answer is pectic methyl esterase. Next question. The green color of green vegetables is lost during heating due to the production of. The green color of green vegetables is lost during heating due to the production of. Options are carotenoids, chlorophylls, pheophytins, and flavonoids. The green color of green vegetables is lost during heating due to the production of option C, pheophytin. That is, we know that chlorophyll pigments are present in all green vegetables, and these chlorophyll molecules are converted into pheophytin by the loss of magnesium ions because of the heat and low pH during processing or something, cooking or something. The green color of green vegetables is lost during heating due to the production of pheophytins, as the chlorophylls are converted into pheophytin by the loss of magnesium ions. Next question. Non-enzymatic browning in fruits is reduced by. Options are sulfur dioxide treatment, vacuum treatment, osmotic dehydration, and all of this. Non-enzymatic browning in fruits is reduced by all of this. That is with sulfur dioxide treatment, vacuum treatment, and osmotic dehydration. That is by avoiding the contact of the surface with oxygen, we can reduce the non-enzymatic browning. So the answer is all of this. Next question. The coffee that may reduce insomnia effect is. The coffee that may reduce insomnia effect is. Options are instant coffee, decaffeinated coffee, regular coffee, and all of this. The answer is the coffee that may reduce insomnia effect is decaffeinated coffee. We know that caffeine is responsible for the insomnia effect. Insomnia means staying asleep or difficulty in sleeping, and this is induced by the caffeine present in the coffee. And if it is decaffeinated or the caffeine is removed, then the insomnia effect is reduced. What is this decaf coffee or decaffeinated coffee? It is the coffee from coffee beans that have had at least 97% of their caffeine is removed. And this can be removed by either by water or organic solvents or with the help of carbon dioxide. That is, the coffee beans are washed in solvent until the caffeine has been extracted. Then the solvent is removed. Then these beans are decaffeinated before they are, or the decaffeinated beans are then roasted and ground. And the nutritive value of this decaf or decaffeinated coffee will be almost identical to the regular coffee apart from the caffeine content. In decaf coffee, the caffeine content will be very much reduced. Next question. The green tea processing does not involve. The green tea processing does not involve. Options are steaming of tea leaves, rolling of leaves, fermentation of leaves, or drying the leaves. The green tea processing does not involve fermentation of leaves. The green tea leaves are often referred to as non-fermented or unfermented teas. That is, the chief manufacturing difference of green tea as from the black tea is the leaves do not undergo any process of fermentation. Next question. Smoking of meat is carried out at about which temperature? That is, the smoking of meat is carried out at about 57 degrees Celsius, 77 degrees Celsius, 87 degrees Celsius, or 97 degrees Celsius. The answer is 57 degrees Celsius. The smoking of meat is carried out at about 57 degrees Celsius. What is the smoking of meat or smoked meat? Smoking is done to preserve the protein-rich foods which would otherwise spoil quickly for a long period. that the smoking is the process of flavoring or browning or cooking or preserving food by exposing it to smoke from burning or smoldering material, most often wood. The meat, fish, and lapsang tea are often smoked. Smoking is done, or it is the process of flavoring, browning, cooking, or preserving food by exposing to smoke from a burning material. And mainly the small meat, fish, and some kind of tea are smoked. Here, the smoking of meat is carried out at about 57 degrees Celsius. Next question. Which of the following is not used for artificial tenderization of meat? Which of the following is not used for artificial tenderization of meat? The answer is sugar. We already discussed about tenderization in this video, and for artificial tenderization of meat, sugar is not used. Rest others, that is salt, enzymes, and electrical stimulation is also used for artificial tenderization of meat. That is for making the collagen more soft and palatable, is known as tenderization. And then next question, homogenization of milk results in. Homogenization of milk results in. Options are decrease in number of fat globules, increase in whiteness of milk, decrease in temperature of milk, and all of the above. What is this homogenization? Homogenization is a mechanical process used to reduce the size of the fat globules in the milk. Homogenization is a mechanical process used to reduce the size of the fat globules in the milk. So, homogenization of milk results in increasing the whiteness of milk. Here the other options are decrease in number of fat globules. It is not true. Homogenization will increase the number, and by reducing the size of the fat globules, it will increase the number of fat globules. And it also increase the whiteness of the milk. How homogenization will increase the whiteness? That is, homogenization, with homogenization, more number of fat globules will scatter more light effectively, which results in decrease in average diameter and an increase in number and surface area of fat globules, which will give more whiteness to the milk. With this, we are winding up this session. Thank you for listening this video. If you like this video, give us a thumbs up. Please subscribe our channel, press the bell button for future notifications, and share this video with your friends and leave your valuable comments. Thank you.